Darwin's main contribution to our understanding of evolution was not in discovering it, but in proposing a mechanism by which it works: natural selection. But natural selection isn't the only way by which a species can change over time. There are other mechanisms of evolution. In this lecture, we'll look at some of them. We'll also learn how to tell if a population is evolving, and then we'll see how that information can be used to infer the mechanism that is driving the changes. As we discussed in the previous lecture, it's populations that evolve, not individuals. This is a subtle but important point. Even if you have an exceptionally successful life, you probably won't alter the future of our species unless, of course, you're Genghis Khan and you leave thousands of offspring behind. Most of us are not quite so fertile, especially females, of course, who have a lot of investment they need to make in order to have a baby. Your traits do affect your ability to survive and reproduce, but the influence of natural selection on Homo sapiens sapiens or any other species is only seen in the change in the population over time. Evolution wouldn't happen if every individual in a species was exactly the same. Individual variability is a prerequisite for natural selection to take effect. And this variability needs to be tied to something that gets passed down from one generation to the next. It has to be hereditary. In Darwin's day, scientific knowledge of genetics was only just beginning, and Darwin himself didn't know about genes. But now we know quite a bit, and we can see natural selection in action even at the smallest scale, what is known as microevolution. Microevolution describes the changes in the frequency of specific variants of genes in a population over the course of several generations. Let's take a minute now to consider the facts of genetic variation. Genetic variation refers to the differences between individuals that are tied to their DNA or genetic code. Not all of our differences are hereditary. For example, body shape does have a genetic component, but life choices can have a huge effect on how someone looks. Our genes are encoded on a set of 23 unique chromosomes, strands of DNA that contain many genes. And we have 2 copies of each chromosome, one from our mother and one from our father, for a total of 46. So we inherit 2 copies of each gene, one from each of our parents. These genes are the blueprints for all of the proteins that we need for our cells to function. These 2 copies of each gene are called alleles, and they can either be the same, so we call them homozygous, or they can be different, heterozygous. When we're looking for evidence of microevolution, we look for changes in the frequency of one allele or another. That is, over generations, does one specific type of gene become more frequent in the population? The interesting thing about microevolution is that natural selection is not the only mechanism by which the allele frequency in a population can change, although it happens to be the one mechanism that regularly leads to an organism's adaptation to its environment. 2 other mechanisms that can change and shift allele frequency are genetic drift, which is caused by random sampling or the effect of chance, and gene flow, or the transfer of alleles between populations. It's not a given that a population can evolve. As we've noted, there are certain conditions like genetic variability that have to be put in place. Well, it turns out that we have a way of assessing the likelihood of whether a population is evolving. It's called the Hardy-Weinberg principle, named after G.H. Hardy and Wilhelm Weinberg, who first demonstrated it mathematically in the early 20th century. The Hardy-Weinberg principle determines the allele frequency in a population that is not evolving. That is, allele frequency should remain constant from generation to generation when a population is not affected by natural selection or other causes of allele frequency shifts, such as genetic drift and gene flow. Once we have the predicted frequency from the Hardy-Weinberg equation, we compare it to what we see in the actual population. If they are different, then we can say that the population is in the process of evolving. When the principle accurately predicts allele frequency from one generation to the next, we assume that the population is in equilibrium. Departure from equilibrium occurs when one of the following 5 conditions is not met. That is, these conditions describe a population whose genes aren't changing, so it's not evolving. First, there can be no mutations. A population remains static so long as alleles aren't altered and genes aren't deleted or duplicated. This condition means that no new mutations should arise spontaneously in the population. Second, the population should engage in random mating. So long as individuals choose their mates randomly, the gene pool remains the same. But if there is a tendency to mate with close relatives, say in the case of inbreeding, or if mates are preferentially chosen from a small subset of the population, then allele frequencies can change. Third, we can't have natural selection. When certain alleles make it more likely that individuals with those genotypes will survive and reproduce, then the gene pool can change. Fourth, the population should be very large. A smaller population is more likely to be affected by changes in allele frequencies simply by chance. This is genetic drift, and it's why when animals are in danger of going extinct, genetic problems become more and more of an issue. And finally, there can be no gene flow. Sometimes individuals who are fertile move into or out of a population, changing the allele frequency and dynamics of that group. We'll talk more about genetic drift and gene flow in the next few minutes. If these conditions of the Hardy-Weinberg equation are met, it means that the population is isolated either geographically, say on an island like Australia, or culturally, say in a hypothetical land where mating with members of a different tribe is strictly forbidden. But these conditions don't seem to describe most of the populations that we're familiar with. And if they're always violated, what's the point of having the equation to begin with? Well, the Hardy-Weinberg equation gives us a starting point to compare our population of interest against. By evaluating which of the conditions are not met and why, we can begin to speculate and eventually prove which mechanisms are causing the change in allele frequency, or the evolution. We know that natural selection, or very roughly, the survival and reproduction of the fittest, is one mechanism by which evolution takes place. But what are the others? Using the Hardy-Weinberg assumptions, we can figure out what other forces cause evolution if we examine which of the assumptions the particular population we're studying has violated. Let's start with the first condition, that mutations do not occur that could change the alleles. Well, we know that's not true for most populations, since mutations do occur frequently and spontaneously, usually as a result of mistakes that happen during DNA replication. You might have heard that miscarriages are most common in the first 3 months of a woman's pregnancy. Well, the vast majority of these pregnancy losses happen because of chromosomal abnormalities. Most of these occur accidentally in the egg or sperm, and many pregnancies end before a woman even misses a period. It almost seems quite remarkable that so many babies are born healthy, given all the things that can go wrong along the way. Of course, if an accidental mutation happens early in the pregnancy and the embryo never develops, that mutation has no chance of being passed on to the next generation. That baby's never born, that baby can't reproduce. But there are also random mutations that occur that are less devastating and don't impede reproduction. You might think that such mutations would have the largest effect on the evolution of a population. A direct change in the genes, after all, seems like the most efficient way to change alleles. But to understand the potential of mutations to alter populations, we first must consider the vast number of genes that form the code of our lives. The human genome contains the blueprints for about 20 to 25,000 genes on our 23 pairs of chromosomes. Now, that seems like a lot of genes when you consider that these spontaneous mutations occur at random. Even if there are many mutations in a population, there are 20,000 genes to choose from, so the mutation rate for any given gene is pretty small. And for this random mutation to have a cumulative effect that's visible across the population, the rate of mutation would have to be pretty significant. So high, in fact, that it's unlikely that the species could still function well with such a high rate of mutations. So how do we reconcile the fact that mutations happen, but that they don't have a major effect on the evolution of a population? Well, these random mutations are in fact an important prerequisite for evolution to be possible. Although mutations aren't a force of evolution in and of themselves, they set the stage upon which other evolutionary mechanisms can act. Because random mutations are, by definition, random, they don't have a big influence on the rate of alleles in a large population. Let's unpack this a bit using an example. Let's say that we have a large population of furry bunnies. Let's make it 100,000 of them. Each of these bunnies has 15,000 genes, and one of these genes codes for fur color. And let's say that the rate of spontaneous mutations in any gene is, let's say 1%. That's pretty significant. But even so, that means that in any given generation, only 1,000 bunnies have a mutated gene. Of these, if the rate of mutations for all genes is the same, the chance that even one bunny would show a mutated fur color is still only 1 in 15. Even if the rate was 10%, there's still a chance that none of the bunnies would show a mutated fur color. Even if several did, the chance that the mutation would be manifested in the same way in a number of animals, significant enough to change the population, is still really small. So mutations don't have a big influence on the gene pool, provided the population is large and the animals, like us, have lots of genes. The second assumption of the Hardy-Weinberg principle that is sometimes broken is that mating is random. Random mating ensures that any mutations that do arise are dispersed among the population rather than concentrated in one group. The negative consequences of non-random mating, as we can see when we breed dogs, for example, to ensure that they are purebred, can be devastating. While purebred dogs are often preferred for their aesthetic qualities, they also have many more health problems than your average mutt. Examples of issues that can arise are sinus problems in dogs like pugs and bulldogs, deafness in King Charles Spaniels, and much more serious issues like higher rates of cancers, heart disease, bone disorders, neurological diseases, and weak immune systems. Purebred dogs live much shorter lives than your average mutt from the SPCA. They are also often not as smart. Why does this happen? Because purebred dogs are bred from two parents within the same relatively small gene pool, and these gene pools are getting increasingly limited, with many breeders using dogs from the same family to generate puppies. Inbreeding. The perils of inbreeding are also not uncommon among humans. Just look at the rates of hemophilia, psychosis, and other genetic diseases among members of royal families. These diseases are more common when relatives mate with each other, because the chances of a child inheriting a bad copy of a gene are greater when there is less variability in the genes of his parents. So familial diseases become more frequent. What if the population that we're studying isn't very large? Violating this assumption can lead to genetic drift, when allele frequency shifts because of random selection effects. To illustrate genetic drift, let's say you have a population of candies. We'll start with a small population of two flavors, blue and white. Let's say that the color of the candy is based on one gene, and each color is a different allele. Now, let's imagine we lived in a jar where the allele frequency starts out even. So for every 10 blue ones, there are 10 white ones. For the sake of simplicity, let's say that it only takes one parent to make an offspring, and that offspring is always the same color as the parent. So imagine randomly choosing candies as parents from the first jar and putting the same color offspring into the second jar. Once you've finished populating the second jar with 20 candies, you'll see that the allele frequency has changed. Because there won't be an even number anymore, there will be slightly more of one than the other, just due to random chance. If you keep doing this over the course of several generations, you'll see that genetic drift happens fairly quickly in small samples. It might even get to the point where one allele is completely absent, extinguishing that variability. So in small populations, genetic drift can have a big influence, decreasing genetic variability and therefore making the population more vulnerable to diseases or extinction very quickly. Genetic drift plagues small populations of endangered species. This is why we worry about numbers of animals of a species and why, when there are only, say, hundreds left, a species is almost certainly doomed. It's because there is less and less variability in the gene pool, making a catastrophic event more likely to wipe out everyone. There just isn't enough variability for natural selection to act upon a gene, or of a familial disease to affect much of the population. Sometimes, if a small group of individuals finds itself isolated from the main pack, its gene pool might differ from the general population simply by chance. Then, as those individuals reproduce, they pass along this specific set of genes, and over time, they become more and more different from the main population. This is a type of genetic drift called the founder effect. We can see the founder effect in action when storms or other forces of nature blow one subset of a species to a new location, say from one island to another in the Pacific Ocean. Or when a species gets introduced to a new environment, say rodents that traveled on ships to Hawaii. But we can also see the founder effect directly in our own species, as it probably explains why certain hereditary illnesses are found in isolated populations of humans, like the island of the colorblind, beautifully described by Dr. Oliver Sacks when he visited Pinjalap in Micronesia, where 5 to 10% of people have achromatopsia, complete color blindness, which is very rare in other parts of the world. This is not your usual red-green confusion seen in many men in particular, but rather a complete absence of color vision because the retina in these individuals has no functional cone cells. We'll talk about cone cells and how we are able to see color through these cones in a later lecture. When the isolation of a subset of individuals from the main population can produce the founder effect, it is also possible that a sudden catastrophic event can drastically reduce the size of a population and also cause a gene pool change. Getting back to our candy jar, it's as if one generation or jar was attacked by a group of hungry school children and only a few candies were left behind. Now, assuming the school children chose randomly, there would still very likely be an overrepresentation of certain alleles in the surviving population. Maybe there are only 3 yellow ones left and 1 red one, simply by chance. This is another kind of genetic drift called the bottleneck effect. The bottleneck effect can be created when a larger population is culled because of some catastrophic event, hungry school children, or an environmental disaster, or the introduction of a new predator. Like how the introduction of zebra mussels native to Asia has wreaked havoc on the native species of fish and plants in the Great Lakes. The outcome is similar to the founder effect. A small proportion of the original population ends up having a much larger effect on the gene pool, but the causes are somewhat different. Population reduction in the case of the bottleneck and population isolation in the case of the founder effect. Both situations are important for us to understand because our actions often cause just such effects in other species. For example, human activity in Illinois during the 19th and 20th century caused much of the prairie land to be converted into farmland and other uses. As a result, the population of prairie chickens vastly decreased from millions to hundreds. Those birds that did survive showed little genetic variability and a hatching rate of less than 50%. To test whether genetic drift and the bottleneck effect caused this major decrease in allele frequency, scientists compared the DNA of living chickens with that of museum specimens collected in the 30s and 60s. Sure enough, the current population had lost many alleles that were present in the museum species. To fix the problem, species from neighboring states were introduced to the Illinois population, thereby making the gene pool more variable. and egg hatching rate improved to 90% as new alleles were added to the gene pool. We have one more mechanism predicted by the Hardy-Weinberg principle to discuss, and that is gene flow, or the transfer of alleles between populations. You'll remember that the last assumption of the Hardy-Weinberg equation is that genes don't flow into and out of a population. That is, individuals from one group don't mate with those from another population. Now, we're not talking about inbreeding here. Remember that in both cases, the populations are very large. For a population to be in equilibrium with allele frequencies remaining constant, which is what the Hardy-Weinberg principle is designed to detect, alleles can't be flowing into and out of a population, as would be the case if individuals were flowing into and out of it and mating with members of different populations. With advances in transportation in the last 100 years or so, we've seen a huge increase in immigration. Individuals from relatively isolated gene pools are now intermingling and mating with each other in our own species. French Polynesians, for example, can now easily travel to faraway places like Brazil or Germany and mate with the locals there. This movement from one gene pool to another is gene flow. But like mutations, migration between populations tends to be fairly random. So the effects of gene flow on our global population are relatively subtle. Just think about human immigration. There are all kinds of people moving between countries, and they are not being sorted out in terms of their alleles. So, like random mutations, the effects of gene flow on allele frequency are fairly low. In fact, they also cause the opposite effect. They can reduce genetic differences between populations, bringing two groups closer together genetically. The more people from a particular part of the world, say China for example, move around and emigrate to other parts of the world, mating with the locals, the fewer differences there will be between Chinese people and people from other countries. Nowadays, immigration is so common around the world that we can almost think of humanity as one big population rather than several isolated groups. Globalization is in full force, and gene flow quietly continues its work. So now that we've considered mechanisms of evolution that are not natural selection, it's time to turn back to Darwin's theory so that we can better understand the impact of natural selection and evaluate his theory for ourselves. I started the last lecture, my introduction to evolution, with a story about the peppered moth in England and its adaptation to a polluted forest. Then I also talked about Darwin's finches in the Galapagos and explained how beak sizes evolved on particular islands as an adaptation to the food most abundantly available in that environment. These two examples illustrate what makes Darwin's mechanism of evolution so different from the other four mechanisms that we talked about in this lecture. Namely, natural selection provides an explanation for the diversity of Mother Nature and the observation that species are precisely adapted to their environments. It's important to understand that evolution by natural selection is not random. Mutations are random, but sorting by natural selection favors some alleles over others simply because the realities of any environment favor the attributes of some organisms over those of others. And that leads us to one key concept in the theory of evolution that I have not yet talked about: fitness. We often hear people describe evolution by natural selection as survival of the fittest. And as such, we imagine two big male gorillas beating their chests at each other with the strongest one gaining the most access to females. But the concept of fitness goes way beyond individuals competing directly against one another, and more often than not, is much more subtle. For one thing, it's important to remember that we're talking about fitness as it relates to the ability to produce more viable offspring. So, competition that occurs after the age of reproduction has no effect on evolution. Unless, of course, it somehow affects the likelihood that the offspring themselves will reproduce. If, for example, having grandparents around makes their grandchildren more likely to make it past childhood because they can help the parents out, and therefore their grandchildren are more likely to reproduce. So we are most interested in relative fitness, That is, how much an individual contributes to the gene pool compared with other individuals. Greater fitness might be demonstrated in individual battles, as in the gorillas mentioned a moment ago, or in the ability of a moth to blend into a tree and thereby avoid being eaten long enough to have babies, or the skill of a barnacle in obtaining a greater proportion of available food, thereby depriving its neighbors of adequate nutrition and preventing them from reproducing. Fitness as a measure is only meaningful if it is relative. For example, the total number of offspring on its own is irrelevant because some species produce more offspring per individual than others. So for example, if I told you that my roommate had 16 children, you'd probably call her a very fit person, at least where evolution is concerned. But if I later revealed that she's actually a common housefly, you'd be less impressed, since houseflies produce about 500 eggs over their lifetimes. Suddenly, a paltry 16 seems completely insufficient. Speaking of offspring, one of Darwin's most original contributions was the description of sexual selection and its effects on a species. Sexual selection refers to the process by which some inherited traits make an individual more likely to find and secure mates. Sexual selection is a subtype of natural selection. A consequence of sexual selection is sexual dimorphism, when the two sexes in a species develop very different characteristics. Peacocks, for example, are known for their colorful plumage, which not only attracts mates, but also makes them easier prey, since they stand out and their long feathers impede the speed with which they can run away. That's why peahens tend to sport earth tones and shorter tails to blend into their environment and escape harm. But peahens choose their mates based on their outfits, so the peacocks have evolved to sacrifice safety for fashion. Of course, traits that are selected for based on sexual selection must also indicate that individuals with those traits have other good genes. That is, that these traits are correlated with desirable heritable characteristics. If they weren't, then the offspring of the showy peacock would quickly be eliminated. Often these traits are indicative of overall better health. So the showy plumage of the peacock indicates that he's healthier than his less showy counterparts. Sexual selection seems to be a particularly potent form of natural selection because it acts directly on reproduction. The ability to survive long enough to reproduce is essential, but if you can't find a mate, you definitely won't be passing on your genes. In today's lecture, we've examined the forces that drive evolutionary change from genetic drift and gene flow to natural selection, which leads to consistent adaptations to specific environments. But why, if we've been evolving for millions of years, are we not perfect yet? The answer is because Mother Nature is a tinkerer, not a designer. Evolution, or selection more specifically, can only act on existing variations in a species. Only alleles which are already in the current population can be selected for. Nature does not create new alleles on demand. If the allele that might save us isn't in the population when a catastrophe hits, we won't survive it. But since mutations happen all the time, and we are part of a very large and diverse population, there's at least a small chance that a few of us, lucky enough to possess those traits, might keep the population going. A related point is that evolution doesn't throw away ancestral anatomy and build new traits from scratch. It co-opts existing characteristics, like refurbishing a computer monitor to turn it into a television rather than building a TV from new parts. Adaptations then are often an inelegant solution to a problem. One might call them a compromise. It's not ideal for our babies to be born in such a state of helplessness. In fact, during the first 3 months after birth, in a period often called the 4th trimester, our babies are still undergoing rapid development and are completely dependent on their caregivers. But if they were born 3 months later, their large heads wouldn't fit through the birth canal, so Mother Nature compromised. Evolution is a product of the interaction between chance, natural selection, and the environment. Each of these forces is a major player, and by understanding how evolution has worked in the past, we can more accurately predict how our actions might affect species in the future. We're living in a particularly volatile time when bioengineering, our environmental impact, and our technology can greatly influence the survival of other organisms and ultimately our own species. We might even be at the cusp of reversing evolution and soon be able to reconstruct extinct animals from bits of their DNA. We are part of nature, but our ability to select for certain traits is unprecedented in the history of the Earth. Understanding evolution is our best tool to prevent catastrophe and to create a better world.